Hi, my name is Xi Mohanty, and I've conducted a study on developing a treatment for nasal squamous cell carcinoma using a type of bacteria by examining the apoptotic properties of Caulobacter crescentus, a type of bacteria. This study is called Utilizing Caulobacter crescentus to Target the BCL2 gene and NF-kappa B transcription factor to cause apoptosis in nasal squamous cell carcinoma. The most common type of paranasal sinus and nasal cavity cancer is squamous cell carcinoma. This type of cancer forms in the thin, flat cells that line the inside of the paranasal sinuses and the nasal cavity. If the cancer is located only in the nasal cavity or paranasal sinus, the 5-year survival rate is 82%. However, if it spreads to nearby tissues or organs, the 5-year survival rate is only 43%. Squamous cell carcinomas of the sinuses represent an aggressive malignancy. Risk factors associated with this cancer are smoking, the HPV infection, and exposure to nickel and welding fumes, which affects many of the world's industrial workers. The Caulobacter crescentus is a type of bacteria that is commonly found in freshwater environments and is adaptable to environments with a low nutrient content where tap water and distilled water can be habitats. The Caulobacter crescentus populations grow in dense communities where fluid flow is present, which shows that these cells must contain attachment mechanisms that promote local and immobile colonization. To maintain an attachment to the surface when subject to hydrodynamic forces, the bacteria utilize multiple adhesive structures, which include a strong adhesive grip at the tip of the polar stalk, and flagella are present at the opposite swarmer cell section. Swarmer cells can be secluded by density centrifugation to produce harmonious cultures that can travel throughout the cell cycle in a synchronous fashion. Swarmer cells transform into stalked cells by releasing a flagella and growing a stalk from the same cell pool. In the swarmer cell, chromosome replication is oppressed but initiates upon the generation of the stalked form. The Caulobacter crescentus is a gram-negative bacteria that is used as a model system for studying bacterial development as well as the cell cycle of bacteria. The creation of a new flagella at the cell pole depends on continuous DNA replication. The utilization of bacteria in cancer therapy takes advantage of various strategies. The use of bacteria in cancer therapy and treatments creates strategies including native bacterial toxicity and the fact that the bacterial agents can control the expression of anti-cancer agents. The utilization of live, reduced, and/or genetically altered bacteria, or in a combination with different agents, has been tested in various studies. The most common and promising bacteria used are Salmonella, Caulobacter, and also Streptococcus. The Caulobacter crescentus has the ability to present proteins and peptides at a high density using the crystalline S layer, which has an important role in the growth and survival of the bacteria. The function of this layer includes enzyme display, maintenance of cell cohesion, and in pathogens, interaction with the host and the immune cell. This provides various opportunities to experiment with the combinations of signaling, targeting, or immune modulation proteins to improve immune effects present in anti-cancer impact to tumors. In the status quo, treatment includes surgery or radiation therapy. Treatment options for stage 1 nasal carcinomas include the surgery for tumors of the septum and radiation therapy for tumors in the superior walls. However, patients who cannot receive surgery or radiation find that their condition worsens. Additionally, surgery and radiation therapy are expensive and invasive, but this bacterial treatment serves as a sustainable solution. Recent studies have shown that stimulating NF-kappa B, a transcription factor, previously thought to be strictly anti-apoptotic, can cause programmed cell death. In this study, I have investigated the mechanism behind how the bacteria impacts the levels of NF-kappa B and BCL2, a well-known transcription factors involved in programmed cell death, and what pathway the bacteria activates this. I performed 3 different assays to measure cytotoxicity, the concentrations of the BCL2 gene and NF-kappa B in both normal squamous cells and cancer cells when exposed to various concentrations of the Calypbacter crescentus bacteria. I hypothesized that the highest concentration of bacteria would have the highest levels of NF-kappa B and the lowest levels of BCL2. In the study, it was found that the NF-kappa B levels were elevated in the cancer cells with higher dosages of bacteria, and the BCL2 levels remained stable. It was also shown that the NF-kappa B and BCL2 levels remained stable during treatment in the healthy cells. These results, however, are still being analyzed, as this study is very recent. This may show that the cancer cells are dying via an inflammatory response such as necroptosis, while leaving the non-cancer cells unaffected. The results show how these organisms can advance knowledge and technology dedicated to creating a safe, non-invasive alternative to the status quo cancer treatment options. Thank you.